All right, for the last 10 minutes or so, we've been talking about the fact that a cell has to make proteins. And those proteins come in different types. And what the proteins do depends on their shape. And how they're shaped depends on which amino acids are combined in which order. And which amino acids are used and in which order depends on DNA. Now, how DNA guides that building, you don't have to worry about right now. But you do have to know that DNA guides that building. But DNA has had that responsibility for 3.8 billion years of Earth life without us understanding it. So now that we understand what DNA does in the cell naturally, humans are beginning to think about what we can do to manipulate DNA to serve our own purposes. And I want to point out two facts that you already know, but they're facts that should be connected. First fact: you are a citizen of the richest, most powerful, most technologically advanced nation in the history of the planet. Second fact: that same nation is a democracy. That matters because so much of the DNA research is done in the United States, and it is the group of American citizens who, at the voting booth, can determine which DNA research should be encouraged by the government, perhaps funded by the government, and what kind should be outlawed. And there are examples of DNA research that some people think should be outlawed. Here is one of my childhood heroes, Michael J. Fox. This is an actor who famously played the role of Marty McFly in a wonderful movie called Back to the Future. This movie's from 1985, but it's totally awesome and every American should see it. So Michael J. Fox here is testifying in front of Congress, trying to get them to increase their funding for a certain type of DNA research. It's really important that the citizens of the United States understand what can and can't be done with DNA in order for our democracy to decide what is best. And everybody is concerned about the health of their babies. And some people may or may not be concerned about genetically engineering birds to have four legs or tomatoes to have long shelf lives at the store. But all these things are possible, and some of them have already been done, and still a lot of people are upset. So we have to know what's up and vote accordingly. Now, something you'll hear me talk about from time to time is that pop culture often misunderstands DNA and talks about it in very strange ways, especially in some of these movies like the Hulk or X-Men and Spider-Man. So, DNA's importance is nicely highlighted by its influence on movies, but obviously those movies are not very accurate in their depiction of how DNA works. So be forewarned and don't pay too careful attention to that. Now, I want to talk a little bit about an episode that's really important in our history of understanding why DNA matters so much. And it goes back to the 1940s. All right, in the 1940s, it had been known that bacteria, one-celled organisms, that they reproduce asexually, meaning without having sex. So one bacterium could produce two offspring by basically splitting itself in half. And we knew that bacteria did not have sex involving two parents. But then, using a microscope, something was observed that really surprised people. And it looked kind of like this. You should definitely take notes. Anything not on your slide, you should get on your slide. So we have one species that has these long protein-like structures called fimbriae. And think of these like biological Velcro. They allow this bacterium to cling onto a surface so that it can parasitize or be a parasite to the host. So fimbriae can cling on to the tissue in a throat, contributing to sore throat. They can cling on to gums in your mouth, giving you gingivitis. They contribute to all sorts of different types of infections. But the idea is that these fimbriae are based on some genetic code that's inside this bacterium. Well, it was observed with a microscope that this bacterium and a different bacterium, a harmless one of a different species, they interacted together in a strange way that hadn't been seen before. They did not have sex. They had no offspring with one another. But an extension of the cell membrane was made to create a sort of bridge between these two bacteria. The bridge is called a pilus. You don't have to know that term. But we later determined that a small chunk of DNA from the harmful bacterium was able to move through the pilus into the harmless bacterium. And later, the harmless bacterium started growing fimbriae. So, why don't you start drawing some fimbriae onto this one? The idea is that this bacterium genetically altered this one without having sex. We had a horizontal gene transfer, a transfer of genes between two organisms without having sex. And that caused this one to become harmful and infectious. This was stunning. We didn't realize that this occurred. And it also made us worry because we saw, oh my gosh, species number one can make species number two harmful. And the antibiotics that we make can be made useless if bacteria are constantly trading genes. It's like trading tips for how to survive the antibiotics we use. So this is it's a troubling idea. But accompanying that fear was a dream. And the dream was, if bacteria can change each other while they're alive by putting genes into each other, could we put genes into bacteria and change them to fit our needs? And this is a dream that has saved the lives of millions of people with type 1 diabetes.